Hi, myself Lalith Mohan. I'm assistant professor working in STC Degree College and I'll be handling Botany. And in this particular class, we will be discussing about the topic called as mechanism of enzyme action. As in our previous class, we have already completed about enzymes, its introduction part and its nomenclature we have completed in our previous class. And this class, we will be discussing about the topic mechanism of enzyme action. So what do you mean by mechanism of enzyme action? In a simplified way, we can say it as how actually an enzyme works. How an enzyme will actually work. So how an enzyme will work? So before understanding how an enzyme will work, we will understand about enzyme. How the enzyme looks. First one, enzyme. So what we are using the enzyme for? Enzyme is used in case of a particular reaction either to decrease the activation energy, or to increase the rate of a reaction. If it is said to be a reaction is about to complete in 20 minutes, to make the reaction complete in a decreased time or to increase the rate of reaction, we will be using a particular enzyme here. So that is the actual function of an enzyme. So how will this enzyme function? How will this enzyme work? For that, we have to understand about the active site. You should understand about active site. So what do you mean by active site? So active site is nothing but a part of an enzyme. Imagine this structure over here is an enzyme. And the structure which is found in this part can be called as well active site. This is the region called as active site. Here, this is the region meant for the attachment of a substrate. This is the region meant for the attachment of a substrate. So what do you mean by substrate? If you want an enzyme to work properly, there should be a combination of two components, namely an substrate as well as an enzyme. An enzyme can work efficiently only when there is a presence of a substrate for it. So let's take an example of cellulose. Cellulose is a common protein which we come across in the cell walls of our plants. To break this cellulose, if you want to break this particular protein cellulose, we require an enzyme called as cellulase enzyme. We require an enzyme called as cellulase. As the S defines here as enzyme. It defines it as an enzyme. So we require an enzyme called as cellulase to break down the protein called as cellulose. How will this cellulose break in cellulose? That is nothing but the substrate. Here the cellulose will act as the substrate and the enzyme is called as cellulase enzyme. When there is a combination of cellulose with that of cellulase enzyme, that can break the protein cellulase into numerous parts. This is the structure. This is the structure of your enzyme and this is the knowledge about your active site. And the next one, we will be discussing about the next important aspect, that is enzyme substrate complex. That is nothing but enzyme substrate complex. So what do you mean by enzyme substrate complex? See, as I told you before here, when this cellulose is combined with that of the cellulase enzyme, these both will combine to form your enzyme substrate complex. This will form your enzyme substrate complex. Once this enzyme substrate complex is formed, that can lead to the formation of your product. That can only lead to the formation of your product here. Let's see a diagrammatic representation of the particular enzyme substrate complex. Imagine this part as your cellulose. This can be called as well cellulose and this will designate it as cellulase. See, this is the enzyme called as cellulase enzyme used to break the protein called as cellulose. This is the cellulose which is acting as a substrate. This is acting as a substrate here and this is your cellulase called as enzyme here. The first step is called as entering of active site. So, if an enzyme has to work, the substrate has to reach near the active site. That is the first step. That is entering to the active site. The second one is called as attachment. Once this substrate enters this active site, it has to attach with that of the enzyme. It has to attach with that of the enzyme here. That will be our second step. And the third step is the formation of enzyme and product complex. Once the attachment is completed, There is a formation of a stage called as enzyme and product complex. There will be a stage called as enzyme and product complex. Following this step will be our the release. So once there is the step completion of this product the step called as E and P, that is the enzyme and product step is completed. After this step we come to a stage where the two products will be released. The product which is formed as a resultant of this enzyme substrate complex will be released in our last step. And again this enzyme will be again ready for the next step, that is again the conversion of your cellulose into cellulase. I am just giving an example of cellulose and cellulase. There are so many enzymes and there are so many proteins which need to be doing the very different functions here. So this is about the mechanism of enzyme action. What do you mean by an enzyme? What do you mean by an active site, and how this enzyme substrate complex will be working. So the next topic we are going to discuss here is the kinetics of enzyme action. In our previous thing we learned about the mechanism, and in this topic we will be learning about kinetics. What are all the kinetics involved in an enzyme substrate complex, or how the enzyme will work. And this was contributed by two great scientists named as Michaelis as well as Menton. These both were awarded for their work about the kinetics of enzyme action. The Michaelis and Menton in the year 1913 they discovered this particular kinetics of enzyme. When they were working on an enzyme invertase, about the catalysis, the hydrolysis of glucose and fructose, they discovered this particular kinetics of enzyme. As when they were working on the enzyme invertase, about catalysis of hydrolysis of glucose and fructose, they discovered this particular kinetics of enzyme action. And they given the equation known as E plus S, you raised to ES, and then to the E to P at three different intervals. This will give to us a complete equation like E plus S, ES, and E plus P at three different intervals. What are those intervals? The first interval is the K1. The first interval is called as what? K1. That is the formation of ES. That is nothing but enzyme substrate complex. When it is called as ES, it is called as enzyme substrate complex. It is meaning that enzyme along with substrate will form your enzyme substrate complex at first interval. It means that along with an enzyme, a substrate will combine to form an ES, that is what? Enzyme substrate complex at first interval. What do you mean the first interval? K1 is equal to constant for formation of enzyme substrate complex. What do you mean by the first interval? It is K1, that is constant for formation of enzyme substrate complex. This is the first interval resulting in the formation of an enzyme substrate complex. And the second interval is called as the formation of E plus P. The second interval is the formation of E plus P, that is designated as K2. That is designated as K2. What do you mean by K2? K2 is nothing but rate of constant for dissociation of ES. Rate of constant for dissociation of ES. So once this enzyme substrate complex is formed, there should be formation of a product. Once this enzyme substrate complex is formed, there should be a formation of a product. Once the product is formed, it should be released. Once the product is formed, it should be released. Hence, there should be a dissociation. And there should be a dissociation, that is rate of constant for dissociation of enzyme substrate complex. This formed enzyme substrate complex should be disassociated for the formation of your product, and that is what second interval called as K2. That is your second interval called as K2, and we stop at the last interval called as K3. That is nothing but the rate of constant for conversion of S to P. That is the rate of constant for conversion of S to P. Once this K2 is completed, there will be formation of your products from the substrate. There will be a production of products from your substrate. That is nothing but your last thing called as K3. And this is the kinetics of enzyme action as demonstrated by Michaelis and Menton in the year 1930.